Today, let us discuss the topic structure and functions of chloroplast. Chloroplast is a green plastid. It is a cytoplasmic organelle containing green chlorophyll pigments and is concerned with photosynthesis. The chloroplasts vary in shape. They are spheroid or oval or discoid in higher plants. They are cup-shaped in Chlamydomonas, spiral and ribbon-shaped in Spirogyra, griddle-shaped in Eulothrix, star-shaped in Zycnema, and in the form of a network in Bodogonium. The size of the chloroplast vary from species to species. In higher plants, it is 4 to 5 microns in length and 1 to 3 microns in thickness. The number of chloroplasts varies from plant to plant. In higher plants, there are 20 to 40 chloroplasts per cell or up to 1000 chloroplasts. The algal cells contain one or few large chloroplasts. Each chloroplast is bounded by two unit membranes, namely an outer membrane and an inner membrane. The two unit membranes are separated by a space known as periplastidial space. Each membrane is about 40 to 60 angstrom in thickness. The membranes are formed of proteins and phospholipids which are organized in the trilamellar pattern. The membranes are typically biomembranes. The inner content of the chloroplast is called matrix or stroma. The matrix is colloidal, transparent, proteinaceous and contains starch grains, osmophilic granules, ribosomes and DNA molecules. In the matrix, there are many closed flattened sacs called thylakoids. The thylakoids are also known as lamellae. Each thylakoid is bounded by a single unit membrane and encloses a cavity called loculus. 10 to 100 thylakoids are arranged one above the other in the form of a stalk of discs. This structure is called a granum. A chloroplast has 40 to 60 grana in its matrix. A granum is composed of 10 to 100 thylakoids. The thylakoid contain the pigments, namely chlorophylls and carotenoids. The grana of the chloroplasts are interconnected by membranes tubules known as stroma thylakoids or frets. They are devoid of the pigments. In algae, the chloroplasts are known as lamellate chloroplasts and the grana are absent. The membranes of thylakoids are formed of layers of proteins and phospholipids. The chlorophyll and carotenoid molecules are arranged in between the protein and phospholipid layers of the thylakoids. Each chlorophyll molecule has a porphyrin ring towards the protein layer and a phytol tail towards the phospholipid layer. The carotenoid molecules are arranged alternating the chlorophyll molecules. The membranes of the thylakoid is a unit membrane. The thylakoid membrane contains a number of minute particles called quantosomes. A quantosome is a photosynthetic unit. A typical quantosome is formed of 230 chlorophylls and 48 carotenoids. The quantosomes absorb quanta of light and are concerned with photochemical act. The chloroplast contains proteins, lipids, carbohydrates, DNA, RNA, carotenoids, chlorophyll and minerals. These chemicals are present in the following ratios. Proteins 35 to 55%, lipids 20 to 30%, carbohydrates variable, chlorophyll 9%, carotenoids 4.5%, RNA 3 to 4%, DNA 0.5%, minerals 0.2%. The chloroplast contains pigments. The pigments are concerned with photosynthesis. So the pigments of chloroplast are called photosynthetic pigments. The photosynthetic pigments are of three types. They are chlorophylls, carotenoids and phycobilins. Chlorophyll is a green pigment present in higher plants and green algae. Each chlorophyll molecule has a porphyrin head formed of magnesium porphyrin and a phytol tail. There are several kinds of chlorophylls, namely chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, etc. Chlorophyll A and B are present in higher plants and algae. Chlorophyll C is found in diatoms and brown algae. Chlorophyll D is present in red algae. Carotenoids are yellow or orange pigments. They are found in all photosynthetic plants along with the chlorophyll. There are two kinds of carotenoids, namely carotins and carotenols. that means the anthophylls. They are insoluble in water. Phycobilins are the accessory pigments found in red algae and blue-green algae. The phycobilin of red algae is called phycoerythrin and that of blue-green algae is called phycocyanin. The existence of DNA in chloroplast was first discovered by Riss and Plant in 1962. The DNA of the chloroplast is circular in shape. The DNA of the chloroplast is concerned with cytoplasmic inheritance, protein synthesis, and division of chloroplasts. The chloroplast contains ribosomes. The ribosomes of chloroplasts are smaller in size. They are 70S type. Polyribosomes also occur in chloroplasts. They are involved in protein synthesis. The chloroplasts are partly independent organelles. They can manufacture some of their proteins with the help of their DNA and ribosomes. New chloroplasts in the cells of higher plants arise from the pre-existing protoplasts. Protoplasts are spherical structures bounded by two unit membranes with stroma inside. The inner membrane gives rise to small vesicles which fuse together to form the thylakoids. Chlorophyll molecules are formed from protochlorophyll molecule in the presence of sunlight. In algae and ferns, the new chloroplasts originate by the division of the pre-existing chloroplasts and they synthesize certain amount of protein. Functions of chloroplasts. Protein synthesis. The chloroplasts contain DNA and ribosomes and they synthesize certain amount of proteins. Cytoplasmic inheritance. Chloroplast DNA also serves as extranuclear genetic material. In Mirabilis gelapa, the chloroplast DNA is involved in the cytoplasmic inheritance of plasmids. Starch storage. Chloroplasts temporarily store starch during the daytime in the pyrenoid. At night, the starch is transferred to regions of growth and storage. Oxygen supply. Chloroplasts utilize carbon dioxide for photosynthesis and release oxygen. This oxygen is used by all animals and plants for respiration. Photosynthesis. The chloroplasts are directly concerned with the reaction of photosynthesis. Photosynthesis is a biological process. It is a vital process. Photosynthesis is a vital biosynthetic process by which chloroplasts of green plants synthesize sugars by using water and carbon dioxide in the presence of sunlight.